break, you have maintained this series, and we have to maintain up to the end of the year.
Yes.
That is up to 30th May. Because from 1st June, next our session will start. So,
Dr. Bipran is doing a very wonderful work.
correct.
Thank you so much, sir.
Yes. So, I will suggest again, all attendee today, 17 attendees are there. All attendee that you also suggest some topics of the research. Definitely Dr. Bipran sir will take a note of it. And one by one, he will take for the discussion. So, today's is the genomics, correct? Research on genomics. And today, research on genomics is the center, heart of the all research. Because we can modify the characteristic today only. I circulated one notice, one news.
Yeah.
Which news?
Diabetes.
That is
Diabetes.
the pancreas secretion.
Diabetes.
In the diabetes, these beta cells are defective. But now the scientists are successful in modifying the beta cells so that whenever they are injected in the body of diabetic patient, they will not be rejected, they will be accepted. And the person will become diabetic free only because of the genomics.
Yeah.
So genomics is a playing wonderful job. And all among the 21 members have joined. Very good. Among the 21 members, definitely 50% have joined to learn what is going on, and 50% might have already conducted experiments in the genomics. So, their experience is helpful to all, and the Prakash Halami sir, Gurudeep Nagpal sir, Dr. Negish Negina is there, Dr. Mazid is there, Dr. Dipali is there, Dr. Sathis is there. All these are PhD holders. They will also focus some light on the genomics. So I request Bipran sir, please proceed for the discussion. Thank you.
Thank you so much, sir, for your wonderful introductory speech. So, once again, I welcome all the participants who joined today's roundtable discussion on the topic microbial genomics. As Deshmukh sir rightly said that this microbial genomics is not just about the reading DNA. So it's about the decoding the life's blueprint to address the most of the real world challenges. So today's discussion is an opportunity for all of us to explore those important topics in the microbial genomics, that how Indian and global research are converging to create a more informed, or you can say the data-driven and the sustainable microbial future. So now I invite all the participants who have research experience in this microbial genomics, or if you want to share any knowledge with the participants. So just unmute yourself and you can share your experience on this particular topic, microbial genomics. I request all the participants. Dr. K. Satish Kumar sir. Hello. Now this platform is open. Anyone who want to share the knowledge or any practical experience or research experience on the microbial genomics. Dr. Kirat sir has also joined.
Yes, sir. Good evening to all.
Good evening, sir.
Yeah, excellent topic. Today's topic is excellent. We can see now that genomics is the key regulator So whatever we are trying to do now, ultimately directing everything towards the regulation of genomics, either in microbial cells or in animal cell or plant cell, everywhere. So wherever we want to introduce some kind of special enzyme, for example, in my personal view, last year we were working on some proteins which were getting those proteins in a specific bacteria, but then we tried to express it in another bacteria for the for the better growth conditions and something like that. So at that time, we had to rely on the genomic purposes. We had to create the oligonucleotide, and then we had to insert that to our suitable cell. So it's very fascinating to. I'm talking about an environmental application of that bacteria. So be it environmental, be it clinical or other industrial purposes, we frequently need this type of genetic manipulations or genomic manipulation. So we have to fetch the genes from a suitable organism, and then we have to introduce it to a suitable organism. Yeah. So that's a small view, and let's see other discussions also.
Thank you so much, sir, for sharing your research insights, sir.
Yeah, thank you.
So here, Dr. J. Negina Vijay
Hello.
Singh. Yes,
Yes,
ma'am.
yes, sir. So my name is Nagina Vijay Singh, and I'm currently working here at Madras Christian College as an assistant professor. So what I personally feel about microbial genomics is that we normally focus so much on microbiology, right? So microbiology basically it involves people study a lot of things in microbiology, but then we give very little focus to genomics, is the genomic part. So even me, when I was doing my master's degree, I happened to do my internship there in CSIRC CMB. That is when I actually got little exposure to genomics and how it all works and things. So what I personally, after that only I started doing a little more research on those topics. How the genetic analysis and genomic studies help us to get a proper results, or how it ends up giving us a how we can rely on those data for our research. So even now, people who are doing their general microbiology courses, they are not very much aware of how we can use genomic data for our studies, how we can incorporate those things. So I feel like we have to include those things even from the UG level, so that people will be little aware of what all these things are. So even in my research, I was using all these things. So previously, sir was addressing about diabetes and all things. So if we can study a little more about probiotics and how it connects with, we have a lot of review articles and even research articles coming up saying that probiotic bacteria help us in diabetes, in controlling all these homeostasis of blood glucose level and all these. So those things directly connect, at the end, where we have to study about the genomics of those microorganisms. So I feel like it's a very good topic for us to discuss, and I also, expect little more where we can learn so much about genomic data and genomic analysis from all the other people who are working in the area. It will be little more helpful for us to get to know about those things. So yes, sir.
Thank you so much, madam, for sharing your research experience. So, Vijay Singh madam has rightly said that from starting from the genomics part, that the first sequencing of the Haemophilus influenza that was in 1995, and now it is need to means include all the means metagenomics, computational bioinformatics, and also to some level the synthetic biology, so that we can, yeah, yes, madam. Hello, Rajia madam.
Hello. Yes, I
Yes,
am
ma'am.
here, sir.
Yeah, you are audible, madam.
Yeah.
Do you want to share something, madam?
Oh, okay. Yeah.
Yes, madam.
Regarding the microbial genomics,
Yes, madam.
uh, I would like to mention few aspects which we can take in the current times when AI is booming. So, uh, can we design some courses and projects leading to AI investigations in microbial genomics? Currently, I am also involved in writing research proposals with international collaborations. So, if we highlight little bit in that direction, we can take further, because it is a multi-institutional project. Research proposals are being uh, written and discussed in a group pattern. So if anybody is willing, they can contact me. And microbial genomics has very wide range of aspects which can be investigated currently, and using computational biology. So this is the right time to go ahead and carry research in this direction.
Yeah, it's a nice one, man. Because AI-powered genomic analysis nowadays it's booming,
Yeah,
and artificial intelligence, if we include in this with machine learning, so
yeah,
which can be used to accelerate this genome annotation, as well as we can also predict the microbial behavior.
right, sir.
And even sometimes, not sometimes, but people are working on this aspect, and they are identifying novel genes, and they
Yes,
also can study the pathway. So this AI will definitely help us in genomic analysis.
yeah.
So,
There is an institute in Sweden,
mhm,
uh, and it is like funded by the government of Sweden, and it is growing intensively, enormously in this particular direction. So
mhm,
if anybody interested, they can go to that website and see how they are going ahead, and what all initiatives they are taking, and how they are involving other biologists in these
uh-huh.
projects.
In Sweden.
Yeah, in
Mhm.
Sweden.
Okay, okay, okay. Even
Yeah,
UAE also working on this genomics, I remember. So they are investing so much of money in genomic infrastructures.
exactly.
Yeah. So
Actually,
we'll check that.
we should take the youngsters in this direction, so
Great.
that we can evolve eventually with them. We will also involve, because we are also learning,
Mm-hmm.
and the youngsters will further lead this ahead.
Mm-hmm. Yeah. Yes, yes.
Yes, Dr. Rajya madam, just one query. Not query, I
Yeah,
want your suggestion that we are all, means most of us are working on the genomics part.
yeah,
So can you suggest some AI tools that at the means beginning level we can use those AI tools for our genomic research or for the
yeah,
data analysis?
yeah. uh,
Can you suggest some AI tools, madam?
okay. So AI tools, first, suppose we are going to study the physiology of a bacteria.
Yes, madam.
We want to investigate certain enzymes in that physiological pathway.
Mm-hmm.
Okay. So what we can do is, we can go ahead and take the KEGG study of that pathway. We can do the KEGG study of the pathway, KEGG, KEGG study of the pathway. And then what we can do is, we can use the AI tool to develop the regulatory network and framework. So in AI tools, you can use the there is a like agent. There is a bio agent. There is a bio agent, and what it does is that you have to submit. It is an AI tool and it is an agent. It will automate everything, automate the task which you want to do, execute. So you write the prompt in that and provide the sequence. For example, enzyme A, B, C, D, E, F, G. So you write the prompt saying that compare following sequences in the microbial genome of this strain and give an outcome related to its, for example, functioning part, like enzyme kinetics. If you want to get the information regarding enzyme kinetics, so you can get exact value of enzyme kinetics, and then and the regulatory network will give you quantitative as well as qualitative information about the pathway. So this agent will allow you to compare different microbial genomes with respect to the regulatory pathway, and they will also draw a conclusion, interpretation based on the quantitative values of all this regulatory network in comparison with each other in different strains. So in this way it can be used, and then we can like validate experimentally in the wet lab experiments by knocking out a 2 or 3 or genes in a particular strain, and do that kind of comparative study in multiple microbial genomes.
Thank you, Rajya madam.
Here, one more, what I
Yes.
use, that is the AlphaFold. That is also the best tool for protein structure prediction. So that also gives you with 3D folding models from amino acids. So it is just, hello.
Yes, sir. Hello, Dr. Majid, you are audible. Just unmute yourself. Dr. B. Samugapriya.
Hello. Yeah.
Yes, sir.
I was talking about the AlphaFold. That is one of the AI tool, which is for molecular docking, also for protein predictions. Can use this one. It's a nice one. As well as one more was there, Swiss dock for molecular docking. That is also the AI tool.
Swift, sir.
Swiss dock. That's for molecular docking.
So
So
and
instead
also,
of
so I
because
just
there
want
was
to add
another
a
tool.
little to it,
Okay.
because as you mentioned, the Swiss stock, we also have a Swiss ADMET where you can actually predict the molecules, pharmacokinetic properties can be predicted using that,
Okay.
where,
Okay.
the bioavailability and then the rule of five, and how it enters into if the druggable molecule can be entered into the blood-brain barrier, all the properties will be given, where you just have to take a specific, even from the GCMS data or something, will be getting different compounds, right? So those compounds can be checked using different databases like, PubChem. And we can just take only the smiles or just a PubChem ID or anything, just copy paste it. Copy it and go to this Swiss ADMET where it's very similar to Swiss doc, same
Swiss dock.
thing.
Okay.
So you just have to go paste it, and once you paste it and click enter, it gives you a complete profiling of how this enters into the body and how all the prediction will be given immediately. It just takes hardly like
What's
1
that?
minute or something.
What is that name? Swiss?
Sir, it's Swiss ADME. ADME.
Admit, sir. Swiss admit.
Yes.
ADMET.
ABET. Okay.
Yes, and from this we can, do for searching for a receptor, protein receptor, we can use AlphaFold as you mentioned. I also still use AlphaFold for it, and There are different tools actually for it, sir. For example, to understand the different variants of the genetic sequence. So we have a deep variant and alpha genome. I have used deep variant, but there are similar tools like that to. It's very similar to AlphaFold also. It also helps in prediction of the structural prediction of a protein, and also it helps in the it helps in understanding the variants in a sequence, DNA sequence.
I came across one more that is a gene farmer. Gene farmer is
Okay.
also there for a gene expression or gene
Okay.
modeling. Yeah.
Okay.
I did not use, but I just saw that
Yes,
website.
yes, yes.
That because it helps. I think it helps in predicting the gene regulations as like that something.
Okay.
Okay, thank you, sir.
But
Thank
Yeah,
you.
it's just
AlphaFold
that
and
we have a lot of tools, but we are not aware of most of the tools that are already present, sir.
yeah.
Yeah, ma'am was mentioning about, Sweden Institute is offering, she mentioned about the collaboration thing. I actually had a little doubt in it. It's just that ma'am mentioned about how we can incorporate AI in studying all these things. We already have certain things like computational tools, and ma'am also mentioned about KEGG pathways, and we also have different databases to study it, and also have different applications like where we can connect, and networking can be cytoscape. Cytoscape is the app where we can actually study the networking of the molecule and all that. So I just want to ask ma'am how we can actually use those AI tools into this study, ma'am. I just want a little clarity on how we can do it, because we already have enough AI is present in present days. We already have enough AIs and we are not aware of it. So how can we actually get to know all these things, and also how we can think little significantly where we can incorporate all these things?
Hello.
That's my question. Yes.
Yeah. Madam, actually, as and when the AI tools are made and like validated, they come in PubMed. Okay.
Yes.
So what is your objective? Based on that, you can search for the AI tools published
Okay.
and validated. Along with the link, it will be there in PubMed.
Okay.
So what is your objective? Like, for example, if you want to target a particular pathway or what you or a particular like particular molecule or a particular product or like function.
Yes, Rajay
Hello.
madam. Yes,
Hello.
madam.
Yes. So if you have any objective in your mind, can you tell me that objective so that we can further elaborate? Madam Vijaya. Hello.
Yes, Dr. Bijay madam is here.
Yeah. Hello, madam.
Yes, ma'am. Sorry. Yes.
So do you have any particular specific objective? Can you tell a specific objective?
Ma'am, this one I actually, I did it even for my PhD. I actually did the same, where I actually take a compound from PubChem, and then I proceeded with all this. I also used KEGG pathways and all that. So my question was, you said, that you're planning to write some collaborative projects
Yes.
regarding this. I just want to know, in what aspects that you're thinking, how we can do. I want to know about that, ma'am.
No, what exactly what you want to do? Any specific objective you want to discuss, or you want to know the procedural details of collaborating? What do you want to know?
Ma'am, even if it is a procedural detail, I want to know, what kind of things can be done in the collaboration is what my question is. So basically,
Okay. See, madam, it will depend on your expertise and the funding agency, the funding institutes expertise. So whatever their mission is, for example, they want to work in the area of drug discovery, then they will expect you to collaborate in the area of drug discovery. And if you have expertise, you can join them. Or
All right, ma'am,
else,
all right. No, I
that's
just
all.
I think I misheard certain things because I actually thought you are actually working on how you can incorporate AI into microbial genomics. Okay, so I was thinking in that aspect. I thought you were telling me in that. So
Yeah.
yes, ma'am. Now I
Yeah.
get it. Yes.
Yeah, you can contact me directly and we can discuss further.
Yes, ma'am. Sure. Yes.
Yeah.
Thank you.
Can I add something here?
Yes, Dr.
Sure.
Madhusudhan.
Yeah. Even with the help of AI, we can design ligands, AI-based ligands also.
Exactly,
So
exactly.
yeah, so that is also because we are working on that. So that also helps. We have so many models, we have so many websites, we have so many softwares, but we are not using that one. So that they have designed. Now we have to work on it. We have to analyze based on those by using those softwares or AI tools. So even in environmental environmental genomics, we can use these AI models so that we can come to know about the what is the microbial roles in carbon cycle as well as the population degrade as what is it pollutant degradation as well as
Exactly.
in soil health. So like that there are so many aspects on which we can work on with the help of AI. So instead of working on like for example if we were using this Autodoc, so it was consuming time, lot of time to adopt the molecules. But nowadays, with the help of SwissDoc and other tools, within few minutes we are able to dock the molecules. So that is
And
what
yeah,
we
I
are.
would like to, sir, I would like to mention one point here that
Mm,
uh, you are absolutely right in stating that these are all virtual
mm,
research, right? So
yes,
once we try to validate the virtual
yes,
research outcomes, we may
correct,
not be close to like 99% or even 90%. So
right.
the best way in yeah regarding the time thing, it is the
Mm,
best way is to look for AI tools based
mm,
on your requirement.
yes, yes,
Even we can develop, like for
yes.
example, I want to there is a like consortia of bacteria, and
Mm,
suppose I want to calculate the growth kinetics, very simple thing, growth kinetics of
mm,
the
mm,
consortium members
mm,
under
mm,
that reactor or a reactor, etc. So
mm,
I can individually do a wet lab experiment. I
mm,
can do the calculations of each growth rate of each bacteria, and that
mm,
data I can use to
yes,
make a AI tool to study the like further
mm,
bioaugmentation and bioremediation of the
correct,
pollutant in that reactor. So
correct.
these are simple tools which for which we need even wet lab experiments. Without
Mm.
doing the wet lab experiments, we cannot be sure about the
Cool, right, correctly.
validity of the AI tool.
Correctly.
Exactly.
Yeah.
Correct.
So
Just
that way,
this only helps to predict so that we can do the further work in wet lab. Uh,
yeah.
yeah. That
And
saves
in
our chemicals, that save our money, everything, time, everything.
exactly, and
Yeah.
same applies in the same thing is being applied in drug discovery nowadays by the
Yes.
big pharmaceutical companies.
Correct.
So now Prasun sir has joined from the SSCT, Sharda University. Are you there, sir, Prasun sir? Dr. Ashok Kumar Lakshman. So, dear students, if you have any query, so we have experts on the microbial genomics. You may ask the, your query by just unmute yourself, or you can write in the chat box. I can take questions from there. Dr. Deepali Bhoye, Gurdeep Singh Nagpal sir, Priyanka Badoria. If you have any query, you may write in the chat box. Because this one is the platform where we can clarify your any doubt. Dr. Prasun sir, are you there, sir? So now we are very fortunate that today with us the Professor Prakash Salami sir is here with us. Hello. Hello, Professor Halami sir.
Yeah, tell me.
Yes, sir. Just request you to share your research insights, sir, and experience.
Yeah, wonderful. Actually, we are doing some work related to the genomics of probiotic bacteria with reference to the functionalities. And also what are the functional genes are there in the probiotic bacteria, like BSH gene is there, EPS gene is there, MOB gene is there. So how they are distributed, what is their number, how they differ from one organism to another, you know. Similarly, in the bifidobacteria, what are the adhesion genes are there, you know. What are the genes which are actually associated with the adhering to the epithelial cell lines, or these genes are common in a different species of the bifidobacteria. And then with reference to the other probiotic functionalities, you know, which actually can contribute the specific health benefits, like no, the other important genes which are actually associated with, as I told, with reference to the functionalities. Other aspect is the looking for the safety of the probiotic cultures, especially with reference to the antibiotic resistance. So this is actually another important area, because if antibiotic resistance, and if it is a transmissible antibiotic resistance, so then it is undesirable, right? So we are actually looking for such antibiotic resistant genes also through the genomics approaches. So we characterize several of our probiotic cultures. Now actually, when you want to bring the probiotics for a commercializations, doing the whole genome sequencing is important. Knowing the genomic architecture is important. Why? Because then you will be able to answer or tell how your culture differs from the existing one, how your culture is unique, how your culture is safe. Does your culture, bacterial culture, has the required probiotic functional genes? You know, all this information you will be able to actually address through the genomic studies. Very important aspect actually we identified through genomic is presence of the antimicrobial substances, no, vectors in production, exopolysaccharide production, vitamin biosynthesis, you know. Now today only I was just listening actually with a seminar around 5P3, 5Ps, you know, how this subject actually is important. And I came to know that the almost 60% population of India is deficient in vitamin B12. And whatever actually we supply through the pharmaceutical product, malabsorption problem is there. Its utilization problem is there. But when we give through the probiotics, you know, then you will not have that a major problem, because these are actually the bioavailable one. And you know that most of this actually probiotic bacteria has the conserved gene sequences for the production of those vitamins, right? Whether be vitamin B12 or B9 or B6 and all these things. So once we see the genomics, the next area of interest is the functional genomics, you know. So gene sequences can be there, but are they really expressing? or those genes are coming into the form of a proteins or the enzymes, or that particular molecules which is actually required for the bacteria to exhibit the health benefits. Okay. So functional genomics is another important area. When we have a more number of the cultures, comparative genomics, because the comparative genomics you can actually identify or able to distinguish how your culture is important or unique or novel, how your culture differs from the existing one. So this information one can easily address or report, and then that other field or areas in this subject, you can actually identify such a specific loci, especially in the lactobacillus plantarum. What we have actually identified is having a more number of mucin binding domains, MBPs, mucin binding domains. And this mucin binding domain actually was a totally new information from our study, where the lactobacillus plantarum from the fermented vegetable origin can actually perform a better, especially with reference to adherence and colonization into the gastrointestinal tract, of the human being. So such information actually being came from our study, and we are continuing those kind of work, as I already told, with reference to the functionality, with reference to the safety, identifying the uniqueness and all. So we actually do the genomics for bacillus species for the probiotic functionalities. Then lactobacillus comprises of lactobacillus plantarum and lactobacillus fermentum. And more importantly, maybe all the audience also will actually will get a totally new information with reference to the bifidobacteria, because our laboratory has a good collection of this bifidobacteria. And for few of the investigators, in fact, we have shared. So our at least the two bifidobacter cultures are available with the whole genome sequence, and those cultures are actually under the safe deposit in NCIM Pune, and the signature sequences are the uniqueness of our culture, has a lot of importance. So the Bilangam whole genome sequencing paper is already published in Archives in Microbiology, and now we have actually communicated the B Brevi. And then B Brevi, actually the genome sequencing also we have accomplished, and we have actually very interesting sequences that we have actually identified. Similarly, this is of course the conventional methods of genome sequencing through high throughput. But what we done for the Bacillus licheniformis was pack bio with the single stretch, So now you can do the wonders, but then how good quality of the sequences you get, So now it is a more a chemistry, it is a more a bioinformatics, and how this you can actually analyze and then utilize for the one or other purpose is actually very important aspect in science. I could actually see some of these colleagues using AI ML tool. They are looking for the advanced field in this genomics area, and they are actually looking for a new collaborative project. So I think it will be actually very wonderful to have such a collaborative efforts in a different food system or the ecosystem to study the microbial genomics. I can go on talking like this way, but I will be actually happy if somebody has some specific questions to answer. I think there are already 4 questions in the chat box. I don't know whether these are the chat. No, they are not exactly the question. They are actually the instruction for the participants. So
One question.
over to you, Dr. Tiwari.
Yes, sir. So just one means I would like to hear from you, sir, that this metagenomics now enables us to study from the individual microbes to the community, means complex community.
Exactly.
If you put some light on the, sir, because this one is your expertise, that human microbiome project.
Yes. So what happened actually, that the very high level of complex microflora exists in our GI tract. Almost initially we thought it is a 500 different species, but now it is more than 1000. And most of them are non-culturable. So these microorganisms how we can actually study what they may be likely doing the purpose, minor microflora, non-culturable microflora. So what we are actually doing through the metagenomic is that we are actually isolating the total DNA sequence and identifying the abundance of those cultures, classifying them in a different phyla and the genus, and then we are actually calculating the diversity. We are calculating the different the species, the number of the different species. We are actually identifying what are the newer species are there, which are not reported. Now, which species is in abundance. So this kind of studies we are doing through the metagenomic. Other than coming to your lab, we are actually going to the sequencing person. So I will not come to Siliguri to cultivate the microorganism. I will go to Kolkata and get the sample sequence done. Sequence means DNA sequence of these microorganisms. And then I will tell you that this much microbial diversity or population is there in a person who is from the northeast, who is from the south, who is from the west. Then analyzing those sequence data with the European or American or Japanese people, I will be able to tell that like a prevetola is there, is abundant in Indian people, but not in the European people. I will be able to tell that Faecalibacterium is more common in a people are those who are actually living in the rural area, but not in the urban area. I will be able to tell that more number of the bacteroids are present in the people, those who are actually eating the vegetarian food over the non-vegetarian food. I will be able to tell that the people with the diabetics has a more abundant the fastidious microorganism than the healthy people with immediately, rather than waiting for the culture data or the petri plate data or the research laboratory data um, about those microorganism. In other words, I hope it was quite clear.
Yes, sir.
Yeah.
Thank you so much, sir.
Yes. Yes.
So I think
Tiwari,
that yes.
Tiwari sir, with your permission, I would like to make an announcement
Sure, ma'am.
whenever, yeah. Okay. Uh, one thing is that recently ANRF has announced the AI in science project proposals, which is going to be a multi-institutional project. 3 years. It will be for 3 years, and the amount will be 50 crores. And it being a multi-institutional, I am looking for collaborators in this area. So if anybody would like to join and discuss.
My laboratory will be happy, madam, from CFTRI.
I'm going to be happy from Madras Christian College. I'll contact you. I already spoke to you.
Oh, wonderful. So Tiwari sir will share my contact details.
Sure, sir. I will share.
Or we can even make a small consortia now only, and then one of them can be coordinated this activities. And then let us see, this can be a very good beginning from the MBSI side, that we met in the MBSI platform and conceptualize this idea, and then we are working together. All right.
Is Dr. Rajya madam, Vijay Singh madam, please
Yes,
fill
sir.
out this feedback form so that we can connect with you by your mail ID.
Yes, yes,
share
sir.
the details, man, so that we will also think of it.
So I think that one question is for Vijay Singh madam, that can Swiss ADME be used in zebrafish studies using the Caribbean quantum dots?
So this Swiss ADME is basically, whatever the data which have been fed on PubChem can be used here to check for, to predict the pharmacokinetic property. So if the compound is present in PubChem, of course you can use it.
Brinda madam, is it clear to you now? Because this Swiss ADME is just prediction for the absorption, distribution, metabolism, and excretion of any compound. Okay. So
Yes, sir. Yes, sir.
yeah.
I didn't got the answer.
Even you can draw the structure by using the different means chem informatics tools, and you can paste there. or as rightly Bijay Singh madam told us that it is available in the PubChem, you can take the PubChem ID.
Okay, sir. Okay, sir. Because I just asked this question for one single thing is that for the admit concept, they are using some kind of drugs and everything. But in my research studies, I'm just using my synthesized quantum dots. Synthesized quantum dots is not an. I have just checked it. It's not in the PubChem and everything. So admit is the one concept I am supposed to do in my research work. So when I saw that Swiss admit is the one thing and admit concept, it just clicked me whether it can be used for my studies also. But if
Ma'am,
at all, yes, ma'am.
yes, sorry to interrupt, but yeah, if you have the structure of your compound, I mean, the structure, right, chemical structure of your compound, you have an
Yes.
option where you can actually draw the structure, nearby you have a column where you can draw the structure, and once you're done with the structure, you can click enter and it will give you. Based on the
Okay.
structure, it can predict all the ADME properties also. So if
Uh,
you know the structure of it, you can draw the structure, it will give you. Even if it is not present in the PubChem, by using the structure, it can predict you. But then you have to be very sure that it is only a prediction. Okay, it's a prediction tool. So you cannot completely rely on the data.
Yes, ma'am. Yes, ma'am. Because for the carbon quantum dots, so I can just say what is the surface functional group, how the structure is, at what kind of quasi-spherical or it is in some kind of what is it, powder state, amorphous or anything. Only that can be recognized from my studies. Until now, they are just still trying to determine the structure of the quantum dots itself. So to check the admit from the structure determination and everything, it was quite tricky for me. So that's why I thought that this will be an easier way. If to determine the structure also, there isn't lots and lots of characterization for me to be done, starting from NMR to everything. But if I gain some kind of structures mix, I will just try to do that, ma'am, as you mentioned.
Yes, 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 sure. And I haven't worked with all these, nanomaterial stuff. So I'll try reading it maybe in the next discussion. If time permits, or if you are there, maybe we can connect. If I get the answers for this, then I'll help you out.
Thank you, ma'am. Thank you, ma'am. That will be so helpful for me. Thank you. Thank you so much.
And there is one not question but suggestion from the Dr. Prasun sir that cloud-based platform for the comparative genomics. I think that this one is for the Rajya madam and the Dr. Majid sir. Is there any cloud-based platform for the comparative genomic studies?
I have no idea of.
Rajya madam. So, any the participants, you have anything, any knowledge or research experience to share, just unmute yourself. For students, if you have any query, you may ask here. So it's my request to all the participants. I shared one feedback form in the chat box. Just fill out this feedback form, and the e-certificates will be issued to all the contributors only.
Uh, yes, sir. You
Yes.
mentioned about the cloud-based thing, right? So I actually have, a cloud-based is basically when I was doing little bioinformatic work, we had an opportunity to use that. Where the MDS, I couldn't use it here in my university where I was studying. So we got access from a different university, and where we actually, got a cloud-based working space where we work through the service, not the direct, computational system, where we use different service for, we created pocket in a server and we connected through, and we were using a different computer from our computer. So that is the only thing that which comes to my mind when I think about, cloud-based work in this area. So that's the only part that I I have little knowledge about it.
Yeah, but very good information actually. A little
Yes.
bit
Yes.
more on the practical orientation was required. Otherwise, very useful for the beginner to understand and
Yes.
clear the doubt. Maybe you could have given what was that example and how many data set you have analyzed and what was the output
So
result on
it's
your own way.
basically yeah, it's basically, I did a molecular dynamic simulation for one compound. We didn't have the provision where high throughput computing system in our university. So I had to rely on outsourcing, but outsourcing was not, that easy for us. So we had a collaboration with the other university where they had high throughput computer. So they actually gave us a server pocket, which was it's just a same user ID and password kind of a thing. You just have to open your laptop and start using those, where you'll get direct access to that high throughput computing, which is presenting far from your place. I mean, miles from your place. So from that, I could run molecular dynamic simulation. I was using Gromax software, but I didn't have such a big RAM for it. So from my 4 GB laptop, I could run a Gromax, which was running exactly on a cloud server.
Yes, you are right, because some of these servers or programs will not be available for everyone, but then they will
Yes,
give you
yes,
the
sir.
link and then access to operate, and then this is how you can actually share your data and get it analyzed, and then you go for the next step of interpretation. So this is actually
Yes,
another growing field in bioinformatics, where the
yes,
large data set, high throughput analysis, use of the quantum computing.
yes,
I hope
yes.
Dr. Tiwari also will appreciate that when I visited in US, quantum computer actually system in Cleveland Clinic, because computer actually works 1000, 10,000 speed higher than the supercomputer. So you can actually
True,
imagine,
true. Yes, sir. Yes,
Tiwari, that before the morning itself your whole analysis is done and you are free from
yes,
the any worries or anything. And
yes,
such
sir.
IBM
Yes,
based quantum systems are very limited available actually right now in the world.
yes,
So there
And
is
now
one
actually it
question.
is a more how correctly you analyze the data, how the bigger data set you actually analyze. What are the different parameters actually you are looking for, and how exactly you actually interpret. I think this is a more important or coming up, whether it is in the microbial genomics or whether it is in a drug discovery or in the medicine or pharma, pharmaceutical science also, or even in the biodiversity studies also, right? This is actually a very growing and very rapidly evolving field.
Hmm.
yes, yes.
But beside going to all this high-tech subject, actually, I was wanted to tell even Dr. Deshmukh that related to even the simple mushroom products or mushroom cultivations, because this area is not covered in our any roundtable discussion.
Sure, sir. I will
Okay.
include.
Yeah, Dr. Tiwari can actually initiate, because we had on the spirulina or blue-green algae previously,
Mhm.
but on the mushroom we have actually not touched this area.
Okay, sir. Sure.
And yeah, everyone actually is actually quite interested to know whether we should eat a fresh mushroom or the preserved or old one, whether we should eat the processed one or the fresh one, you know, or whether we should use what type of mushroom one should consume, how the mushroom actually is useful from our nutrition perspective and all. Even that field, that actually topic can be brought for one or other roundtable discussion.
Sure,
This
sir.
is
Next
a
week we
suggestion
will
from my
try.
side.
Thank you for suggestion, sir. We will try it from the next week, sir.
Wonderful.
So one more question from the Chaitrali Pathak. I am a doubt regarding MLST sequencing. I wanted to perform MLST of Glutamicibacter, but there are no references available. I had tried using primers which are generally used but does not match. How can I go ahead? So this question is regarding the MLST sequencing.
This question is from whom and for whom? From whom and for who?
For all experts, Chetrali Pathak hai.
Hello, sir.
Hello.
Sir,
Chaitanya
I had
Pathak only should tell what exactly she wants to do, I guess. Or
actually,
what
sir,
in what context you are asking this question. Then we will get a
I'm
clear idea.
actually, sir, I am a PhD student, and as a part of my ongoing research, I wanted to perform a multi-locus sequencing of
Yes,
housekeeping gene. But
exactly.
the genus which I working on is not explored yet, or the sequence is not available in database. So how
कौन
can
सा
I
vector है?
sorry, sir?
Which microorganism you are talking about?
Glutamicibacter, which was a previous member of Arthrobacter.
Uh-huh.
So how can we proceed in case of uh, 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 isolate which is not in database? I had tried using universal primers, which is generally used for that housekeeping gene for sequencing purpose. But this does not show the band using this primer.
Are you talking about Corynebacterium glutamicum or this is something different?
Different, sir. I'm not calling bacterium.
Mm-hmm. No, when you don't have sequence, you need not to go for multi-locus typing. You can just have a one or two housekeeping genes. Multi-locus typing actually is needed when one or two genes they don't answer. They show the similarity. So especially you will see this kind of problem in lactic acid bacteria, very high diversity. You will find this problem in Bacillus because Bacillus cereus, Bacillus anthracis, and Thuringiensis, they are
Yes,
actually
sir.
phylogenetically intermixed. Just to give you an example,
Okay.
unless otherwise you check the virulent genes, all other cystineas or any other housekeeping genes, they are all are similar. They can differentiate this Bacillus species only by the virulent genes, or you have to have a multi-locus typing. So when the culture is new, not many sequences are available. Even cystineas will be able to answer, unless and otherwise it should not have an
Yes,
issue
sir.
with the Helicobacter.
Sir, I had already performed a 16 sequencing, but it shows only 94% sequence similarity with Glutamicibacter creatulolyticus. So that's why I had proceed with MLST sequencing, but it does not show any result. Hello, is I
Yes,
audible, sir?
yes, yes, yes.
Yes, madam. Fine.
We are also trying to understand the difficulty which you are facing.
Yes, sir.
You are right to Dr. Tiwari, he will analyze and tell you within a week.
Okay, sir. Thank you, sir.
Or any other bioinformatics person who is here, they can also actually help her. Where you are doing PhD, by the way?
In Baramati, TC College.
अच्छा, तो पुणे में तो मिल जाएंगे ना तुमको लोग, पुणे यूनिवर्सिटी में माइक्रोबायोलॉजी डिपार्टमेंट. No, the approach which you are using is not correct. No, you don't need a multi-locus typing since your culture is unique, your culture is new. Not much sequences are available, so you don't need a multi-locus. Is it clear what I'm trying to tell? Pathak, can you hear
Hello.
me?
Sorry, sir. I had lost my connection, but now I can hear you.
Yeah, you can actually contact any bioinformatics person who is actually open and will be able to address your question. What I wanted to tell was, since your
Yes, sir.
culture is new, not many sequences are available in the gene bank, right?
Yes, sir.
So you don't need
Yes.
a multi-locus typing. I already
Okay.
gave you the example when we need the multi-locus typing.
Yeah, right, sir.
Okay, so you just do the 16 years, or you can say check perhaps the some of one of this hit shock protein or RNA, no RNA polymerase genes, right? Just see in the
Yes.
essential gene, more than one. That will actually answer your question. Just two sequences.
Okay. Sir, I had already performed the MLST, but can I conclude the result with this? I had used that as primer, but does not show the matches with any of gene. So
Primer
can
क्या
I
करेगा
conclude?
if you design primer based on some other culture sequences, right?
Yes, sir.
So not necessarily that those sequences, similar sequences are there in your culture. So if there are no similar sequences, if homology is not there, you will not get the amplification. That means your culture is can be unique, new or different.
Yes, sir.
तो उसमें तुमको पीसीआर नहीं मिलेगा, पीसीआर नहीं मिलेगा तो तुम सीक्वेंसिंग ही नहीं कर सकते हो.
Right, sir.
Yes.
That is the problem actually.
So right now you do first the 16 years, try to get the full length 16 years. Then you take one more some, I think RNA polymerase, one of these gene. I think two sequences are more than enough for you to answer and convince your guide and even publish your initial part of the study. To
So,
tell what is the
ha,
taxonomy of your culture.
yes, sir. I had already performed the 16 sequencing. I am having that sequence with me, and I
Mhm.
had already submitted with ENCBM.
Okay.
So, this is a novel pieces. So I want to publish it. So can I publish it with only the 16 sequencing, or I need to perform a whole genome sequencing?
Whole genome right now is not needed if you have some other cultural, means other functional data. What this culture is, which metabolite it is producing, for what purpose you are isolated, how different the taxonomy is, and don't look very high impact factor journal, medium, basic
Okay.
microbiology, or
Yes, yes,
journal of basic microbiology, or journal of applied microbiology, or literacy in applied microbiology, you know.
yes, sir. I had performed the polyphasic analysis, that is biochemical, microscopic, and
Yeah, that is what through all this test, if you can show that your culture really is a unique, new,
yes,
mean
yes, yes.
with a positive and negative control. You understand?
Yes, sir.
Tiwari Saab will be regularly conducting several such a training program. You ask him separately, he will answer your question immediately.
Okay, sir. Okay.
Hmm.
Thank you.
Yeah, looks quite very important. I hope you could convince what I am trying to tell.
Yes, sir. Yes, sir.
All the best.
It was really informative, and thank you so much for your guidance, sir.
Yes, yes.
Thank you.
And sir, I just wanted to convey some concern of research student which is facing currently during their studies. Sir, due to lack of funds, we are unable to do our work with. I mean, I had lots of culture, and many more of my colleagues also had lots of culture, which might be novel, but we cannot publish it due to we cannot perform the 16 sequencing or identify all this culture. So I think it will be better if we will get some financial help or any other help, so we can um, I mean we can do our work in more efficiently. Hello.
Now, what you do is whatever the data you have, you try to publish that. Once
Yes, sir.
your publication no, appears, people will you will become famous. And then
Yeah.
you try to participate in one other conference, and then tell that time you tell that you have many such cultures, you know. And
Yeah. Yes.
then people
Uh,
only will come to you and will request you to share the culture, and they wants to take up the further work. You
yes,
understand?
sir.
So
Actually, sir, I asked.
you have to actually come in limelight. So
Uh,
MBSI is a one such a platform to come in limelight. You ask Tiwari,
yes, sir.
we also through MBSI platform only we came somehow in the limelight.
Yes, sir. Actually, I asked this question because I had sequence 12 culture, and out of this, six are showing less similarity. That is less than the 95% sequence similarity, which
Mhm.
is already present, the already existing bacteria or genus. So I thought if I sequence more culture, I will get more novel bacteria. No?
Definitely. You are absolutely right. Now, what you have to
Due
do is
to
you store all those cultures properly. You should know how to
yes,
make
sir.
a stock culture and preserve. Because now
Yes,
you may be finding difficulty as you're telling without minimum fellowship and without financial support, you are doing this one. but then
Yes,
you
sir.
store all these cultures. You need not to go back again, and then whatever you have the data, you try to publish that in some SCI
Yes, sir.
impact factor journal. Let the world knows that you have such experience and expertise. You understood? And then
Yes, sir.
whether the sequencing or the biodiversity study that madam previously, what she was talking, initially, you can actually
Yeah.
take contact of her,
Yes,
get
sir.
in
Sure.
touch, and then don't give a free of cost this culture to anyone. Okay, you
Right, sir.
make use of this the raw material, because this is actually a gold mine actually with you, but they should be really actually useful culture. We can't just go on collecting and then storing them without purpose. You understood? They should be producing
Yes.
some good metabolite, or they should be the new than the existing one, the first time isolated. There are several other properties. You understood? It should
Yes,
be
sir.
really the unique or the novel. So you bring such information, make some presentation somewhere,
Yeah. Yeah. Sure, sir.
you will automatically get recognized.
Yeah.
अभी तक कुछ
हाँ,
पेपर वगैरह पब्लिश किया है क्या?
अभी कर रही वो प्रोसेस चल रहा है सर.
तो करना है ना, गाइड को बोलना है कि सर हमको पेपर पब्लिश करना है,
हाँ सर, वो चल रहा है अभी. वो
हम्म।
प्रोसेस में है. माइक्रोबायोलॉजी सोसाइटी का जर्नल है, इंटरनेशनल जर्नल ऑफ सिस्टमेटिक एंड इवोल्यूशनरी माइक्रोबायोलॉजी में.
वो तो बहुत अच्छा जर्नल है, जर्मनी से पब्लिश होता है वो.
वॉइस वाज ब्रेक.
Yes, yes, all the new culture data,
Recorded.
they will actually newly identified cultures, paper, especially with
Yes,
reference
yes.
to taxonomy, they are actually accepting in that journal. We have some 2-3 papers where we have described especially their probiotic functionalities, with relation to the MUB domains, mucin binding domains, and all these properties we have described. It's a very good journal. If it is your
Yes, sir.
really new and then novel and has industrial relevance,
Yeah.
a lot of importance is there.
Actually, I had I'm working in biocement. So I had first time I used this glutamicibacter in biocementation. So
Uh-huh.
this might be the first report in from India. Uh, so I hope that this will get published in the journal.
हो जाएगा, हो जाएगा. Good luck.
Okay. Okay. Thank you so much, sir.
Thank you, Chaturali and for Alami sir. So, sir, there is one more query from the Dr. Humera. Is if there any possibility of collaboration with the private college faculty interested for AI in science project which Prakash sir was discussing? I think that this question for both Dr. Rajia madam and for the Prakash Alami sir.
Raja madam can answer, I think. I am only talking, so let other people, other audience, other expert also get chance.
Rajia madam.
Dr. Humera, you can unmute
Good evening,
and
sir. Yes, sir. Yes, sir. I'm in, sir. Sir, I just wanted to add one more thing. If there is any possibility with a private college or a private lab, people working with private college or a private lab, is there any possibility of collaboration for AI in science project? Because we
Very
are
much is
recently.
possible, but finally it is the funding, no?
Yes,
Funding
sir.
requirement. And then who is going to support? And then the infrastructure facilities also. So everything is associated, because now the research, actually doing research is becoming expensive.
Yes, sir. We were
Only
discussing
the
about
cheapest thing is
ANRF,
the
sir.
online
That one.
meeting is the cheapest thing, I think, in the world right now.
Yes, sir. So there was a discussion about ANRF project initiative. So that
Yeah,
was my
but
query, sir.
ANRF actually very difficult to get those funding, because the experts they really are expert in something else. They are actually are less bothered to encourage new ideas and actually identify the gap and select the G9 project.
Okay.
Very actually very selected project they funds. I don't know what are actually this criteria. Advertisements are there. Now just a Byrax advertisement also came. One more advertisement is there, I think, in today's paper. But these projects are getting is not that easy.
Sir, is there any criteria that we have to write on so and so lines? Is there something like that? A thin line is drawn so that our project will be accepted. Is there something like that we have to follow?
We are only actually struggling to get the funds. 2 projects are sanctioned, but we don't know where the money is. Everyone is telling with funding agency, no money. Now I am only actually co-chairman of your Bio E3 for functional food. So you can send it to my group. Maybe if
Okay.
everything permits, maybe you can be lucky. Of course, we
Okay.
have not yet met because I am now newly appointed co-chairman for Bio E3 of DBT.
Okay, sir.
Yes.
Thank you so much, sir.
So, so this is the, I'm taking the last question from the Gurdeep Singh Nagpal, sir. Would like some discussion on soil microbes. By using pesticides, insecticides, the CFU has depleted very low. How to multiply them to increase soil fertility, which is very low for cultivation? Farming sector and farmers are suffering. Our food quality and quantity also very low. I think that you are the right person to answer this one. Prakash sir.
एक बार बोलना पड़ेगा. What was the exact question? I was actually
Gurdeep sir, are you there, sir? Gurdeep sir.
Can you read once again?
Yes. Would like some discussion on soil microbes. By
Okay.
using pesticides, insecticides, the
Yes.
CFU has depleted very low.
Yes.
How to multiply them to increase soil fertility, which is very low for cultivation?
Exactly.
Farming sector and farmers are suffering. Our food quality and quantity are also very low.
See, now Hymedia itself has a research institute on soil microbiome in Nagpur. You know, Dr. Hemant Purohit, he is actually the R&D in charge. And when we had MBSI first conference in Nagpur, he was a invited speaker there. And he said that beside the soil microbiome, they are actually incorporating like a probiotics. Probiotics means not which we are actually consuming, but that will actually improve the soil health, soil fertility. Such microflora also they are developing and incorporating in the soil for a soil health. So such extensive study through the genomics, metagenomics, culturable methods, identifying those particular microorganisms, studying their properties, all these things are needed. And as I told, Dr. Purohit is actually heading such team. Dr. Hemant Purohit, who was actually the head and chief scientist in near Nagpur. Now, after his superannuation, he is actually working for the High Media Research Lab in Nagpur. So those who are interested, they can try to contact, or such work is going on already. What I'm trying to tell, such microbial inoculum pertaining to increasing the soil fertility, pertaining to increasing the microbiome, soil microbiome, pertaining to improving the microflora, modulating the microflora, just like how we actually consume the probiotics. So such probiotics for the soil also, they are developing. What is the present status? Even I am also not aware because our area of research is different little bit here. I hope I could answer to some extent this question.
Yes, sir.
Wonderful.
Thank you, sir. So, dear participants, as we come to the conclusion today's roundtable discussion on microbial genomics, so I would like to extend my heartfelt thanks to each one of you for your active participation, your insightful contribution, and your engagement throughout the session. My special thanks to our today's expert, Dr. Halami sir, Dr. Vijay Singh madam, Dr. Majid sir, Dr. Rajia madam. So I especially thank all the members, students, and colleagues of the Microbiology Society India for making this session meaningful and collaborative. So thank you once again for your time and contribution, and I look forward to meeting you all in our next session. So thank you everyone for joining today's session.
Thank you.
Thank you. Good night. Thank you.
Good night.
Thank you, sir.
Thank you, ma'am.